Hello, thin layer chromatography. This topic we have been talking for the last four lectures. We have talked about the principle of thin layer chromatography, components of the thin layer chromatography system as the stationary phase, the support, and the mobile phase. What is the stationary phase? How to prepare it? How to select it? Depending on the purpose, whether it is partition or adsorption chromatography, whether it is for partitioning or whether it is for separation, whether it is for qualitative analysis, whether it is for quantitative analysis, or whether it is preparative type of chromatography. Depending on that, what kind of the support matrix, what kind of the stationary phase is to be selected. Similarly, based on the nature of the analytes to be separated from the mixture, you have to select the appropriate solvent system. So, how to select correct solvent system, that also is being taught. Also, we have talked about the separation principle by TLC chromatography, and in this context, we have talked about partition coefficient. Once the TLC plate is prepared, of course, ready-made plates are also available. We have talked about that as well. Once the TLC plate is prepared, you have loaded the sample either manually or with the applicator or the auto-controlled applicator. You have to let the mobile phase get dried from the plate. The mobile phase is to the one which is used for solubilizing your sample mixture that needs to be dried, and then this plate is immersed into pre-incubated equilibrated TLC chamber that is filled with the solvent and is allowed to stand till the gaseous phase gets equilibrated. The gaseous phase is generated from the vapors produced by the solvent system. So there is no plain air in the chamber, but it is the saturated chamber, a mixture of the gas phase of the solvents. How it is important, why it is to be done, all these things we have learnt in details. Once the TLC run is done, now the turn is visualize the spots, the analytes that are separated. So in this context, today's lecture we are going to focus on. So this is about the TLC plate, how it would look like. This is what we want as an end result. The TLC plate has completely developed, the colored spots are seen, each analyte is properly separated, and you measure the distance traveled by each analyte and the distance traveled by the solvent front and calculate RF value. But for this purpose, the analyte should be seen as a colored product, right? It is to be identified. So, organic compounds, most of them are colorless, and even if some of them are colored, the quantity that is applied onto the TLC plate is so less that it is not really possible to visualize the colored spots of these biological or organic compounds which are present in the sample mixture. So, what we need to do is you have to choose a visualization method. The visualization method could be non-destructive method wherein the sample analyte is not going to be destroyed, degraded, or reacting with any other chemical reagent. It will retain its natural composition and we are going to yet visualize it. So, compound is going to remain unchanged after the non destructive visualization treatment. But is there any example of this? Yes, exposure to UV light, for example, which is going to give visualization of the spots which absorb in the UV range, and it is not going to interfere with the molecular nature of the analyte. The second type of visualization method is destructive, wherein the compound of interest is converted into something new. So you have to have a process which is going to contain the reagents that are going to react with the analyte and give you a colored complex. Now, when it is about development of TLC plate for visualization, we have to take overview of different methods that are available. So as I said earlier, when the chromatography plate is removed from the chamber, it is to be allowed. to dry either by simple air drying or by oven drying so that all the mobile phase is removed. Then to visualize the zones, you have to apply the visualization method. So colored substances can be viewed by daylight without any treatment, but biological molecules are to be detected using various methods. So as I said earlier, it can be as simple as exposure to UV light. So those which have absorption in the short wave ultraviolets, they can be identified by this method. Alternatively, these analytes, if they can be excited to produce fluorescence, we can expose them to short wavelength. When that absorbing that wavelength, the analyte will go into its excited state, and while coming back to the ground state, it will fluoresce. It will emit light, and that long wavelength can be detected. So this can be another method. Or third is you are adding some chromogenic reagent which will produce the color zone. Or you can add a fluorogenic reagent that will produce the fluorescent zones. Or maybe enzymatic method can be used. Now, these fluorogenic agents also can be added into the gel itself or before the TLC is run. It can be part of it. And when such fluorochrome is there in the TLC plate kept under UV will give the fluorescence and the spots will appear as dark spots against that. So certain destructive and non-destructive methods we will learn as examples. Now here is the first plate wherein we have kept that the plate under the UV and it is fluorescing and the dark spots are the spots of the analyte. Of course, we can have chromogenic sprays or chromogenic chambers. In chromogenic chamber, the best and the simple most example is that of iodine chamber, which is going to non-selectively react mm, uh, with is not interact, not react, interact with the bioorganic molecules and give you brown spots is iodine. So iodine crystals are filled in the chamber, allowed to saturate with iodine vapors, and this TLC plate, after it is dried, is to be immersed in the iodine chamber. And the chamber is to be closed. As iodine reacts with these organic molecules, it will give us brown spots as they are seen in the second plate. Third and fourth plate, you have selective reagents which are selected for the specific analyte. So take an example. If you have glycine conjugated analytes, so a selective agent that will react with glycine is para-dimethyl aminobenzaldehyde. It will give red-orange color with that. Or you can use potassium dichromate and silver nitrate to detect the analyte having sulfur compound. Or you can use ninhydrin for detection of amino acid peptides as well as to detect glutathione conjugates. So selectively, you have to decide the spray agent which will react with your analyte and give the specific colors. Here are some common examples of various visualizing agents like iodine, which is a temporary stain. It is not going to react but interact temporarily with the molecules and give the brown spots. Then you have selective agents for carbohydrates, you have bromocresol green for carboxylic acids, etc. Of course, there are certain non-specific and wide application reagents like charring agent, which includes ethanol and sulfuric acid, and that mixture is going to interact with the and react with the biological molecules, wherein the biological molecules are going to undergo charring reaction, and dark spots of the analyte will develop after this charring agent is applied, followed by heating of the plate. So there are reagents which require heat treatment also, as against bromocresol green, which is used for carboxylic acid detection. You don't need any heating treatment. Again, the analytes are lost forever because you have interacted them with some other chemical reagent and new complex is formed. So these are the examples of destructive method against that of iodine, which is going to temporarily react with it. However, as you take the slides out and keep them in the open air, iodine is going to go into vapor phase and the sample analyte will remain on the TLC plate. So it is also example of non-destructive method of visualization. Now here are certain other ways wherein you are going to make use of radioactive isotopes to detect the analytes separated by TLC. So radioactive analytes we know are used in biology. They include tritium and C14 and so on. They can be used to determine the distribution of substances in a reaction mixture, to find out metabolic pathways of drugs, or to detect the presence of a particular pollutant, pesticide, or any specific component in a given natural sample or tissue or plant part, etc. So, these radioisotopes can also be used as tracers for analytes separated by TLC. So, for the detection of radioactive substances on TLC plate, liquid scintillation is one of the methods that is used. Now, second method is autoradiography. So, and third is the radio TC, TLC scanner is the instrument which is used based on either of these principles. Now, here in this diagram, as you see, the scintillation array with the reflector is there. The TLC plate is kept on this, and the photodiode array is one of the labels here. Now, whatever the emissions are given by radioisotope, which is present along with the analyte here, will hit these sensors. The signal will be multiplied in its amplitude by photodiode arrow, and the multiplied or intensified signal will be received, and that will be converted into a digital display which will come in the form of chromatogram, the graphical representation of the analytes present on the TLC plate depending upon the distance from the origin. You will be getting these. Of course, autoradiography is another important technique. Third, just to take a summary of it, this is your TLC plate, this is the applicator, you have loaded the sample, then it is kept into the development chamber, the TLC run is done, the plate is taken out, it is dried, and then it is scanned with the radio TLC tracer. There again, you are going to get such chromatograms generated, which are going to give you different details about the analyte, including the intensity, the quantity, intensity of radiation will talk about the quantity of the analyte. So the graph will give us whether the analytes are properly separated, if they are giving you properly separated peaks, what is the size of the peak, the area under the peak will talk about relative concentration of the analyte in the sample, and so on. So radio tracer technique is one of the methods which is used for visualizing the sample analytes that are separated by TLC. Now, in situ chemical reactions, as we have seen the very first plate, that is application of ninhydrin to detect the presence of amino acids in the mixture. Similarly, these in situ chemical reactions, once done, the product formed can be studied or analyzed using the scanner techniques. So, for example, spectroscopic methods are there, which are going to give us a lot of information about the analyte. So, in situ reactions in TLC plate can be done by various chemical methods. The basic principle of chemistry, like oxidation, reduction, or phytochemical reactions, isomerization, halogenation, catalytic hydrogenation, etc. etc. You can convert your analyte into some derivative form, and then that plate is studied by scanning using spectroscopic methods. Now, next to spectroscopic method, here is another thing where you are going to use densitometers. So it is again an example of in situ measurement method. So as you see, these densitometers are the instruments where you have a, slab to insert your TLC plate on the stand, or on the stage you insert your TLC plate, and it will be scanned, and such chromatographs can be generated. To visualize your analyte, you can take help of densitometers. So light scanning densitometry is the technique used wherein a beam of light is allowed to pass at a particular angle it falls onto the slide. It is allowed to pass through a rectangular slit so that a specific small quantity of beam of light is selected and allowed to fall onto the sample. And optical densitometric scanner is going to uh, produce the data sheet, which is going to be useful to understand the different types of analytes present in it in terms of their quantity. The amount of light absorbed or the amount of fluorescence generated is going to be proportional to the quantity. So the absorbance or fluorescence of the separated zones in the chromatograph can be tracked using an optical densitometric scanner. So, the scanner will give you the visual images. The documentation of the TLC plate can be done using these instruments. The working principle is densitometry. Now, the point is, as the analyte quantities are very minor in the TLC plate, uh, resolution may be compromised when you are going with densitometric methods. So, to get better resolution, TLC systems are coupled with CCD, that is charge coupled devices. So, they are something like your photographic cameras, and they are going to click the image of the photographic plate, and then you are going to uh, convert it into the graphical presentations, and the data can be obtained. So it recently been demonstrated with UV visible absorption, near IR absorption, fluorescence, phosphorescence, and different spots at a time can be scanned because you are taking its image, and which is going to be a very, very sensitive technique. Now, here is another technique which is used to detect very small quantities of analytes present. Of course, it is coupled with radioisotopes again. Not necessarily always it should be for radioisotope. Of course, it can be any visualization treatment, including radioisotope or a fluorescence material. How to handle the TLC plate that can be seen in this diagram. So this is your TLC plate. It is run, it is dried, and it is put onto the visualization system to identify the location of the spot on the TLC plate. And then you are going to take it to a UV visible scanner or different types of scanning system that we talked till now, and such graphical representation can come. But second alternative would be you extract this particular analyte from the TLC plate and then go for its analysis. So for extraction, the commonly used for method is scraping off the spot from the TLC plate and extracting it into a solvent system. But there are chances that some of the analytes, molecules or material may get lost, or some of the support matrices may come along with it, and that will interfere with the system, and additional steps are required to remove the support matrix and so on and so forth. A modification has appeared wherein the sample is not mechanically scrapped alone. However, it is removed with the help of vacuum. So a plastic tube is attached over here near the spot, and the vacuum pump is attached here. It is going to suck the sample like it is working in case of, say, vacuum cleaners in the home, right? And it will be collecting it in the dust bag. So similarly, here the sample will be collected in the glass tube or glass sample holder, and to that you are going to add the solvent system, mix with it. Of course, what about the support material? Is it not present in it? Yes, it is taken care of, because when it is entering the collector tube plate, there is some filter placed which is going to selectively remove the support materials based on the molecular size, particle size. And mostly what you get it in the analysis glass tube is mostly the pure analyte. Then you are going to resuspend it in the solvent system, mix it, and then you can take it for further analyzer. So high resolution imaging based readouts systems, they are combined with such tools wherein you are going to go for efficient extraction of analyte from the TLC plate and go for its analysis. Now next is Yes, when we talk about densitometric analysis, here is one example that I want to share with you is TLC separation of carotenoid pigments from the Tagetes erecta, that is the common marigold flower, is done, and thus carotenoids need to be separated. So the system, solvent system chosen is methanol, acetone, and hexane, 1 is to 1 is to 1. And the stationary phase which is chosen is reverse phase C18, because all these are hydrophobic entities, and selectively silica gel coupled with reverse phase C18, that is the stationary phase chosen. So all these types of supports matrices we have discussed in our previous videos and lectures. So the plate height which is chosen is just 10 centimeters, and you are allowing the system to run once uh, the sample front has uh, uh, the mobile front has run to the other end of the uh, um, plate. You take this TLC plate out, allow it to go for detection. Of course, scanning in absorption or reflectance mode at 450 nanometers that is used, but what you see it as the data is the densitogram that is seen. So when you are seeing it, it can be connected to the instruments which will give you graphical data related to that. So very well you can see here lutein and lycopene and beta carotene and lutein esters, they are separated. So very sharp picks of certain analytes are there, including beta carotene, and here is a mixture of lutein esters which are not properly separated. So TLC scanner will uh, help us to check the presence of different analytes, quantify the analytes based on the densitometric data, and also mm, uh, to give you an idea about, based on the graphical representation, will give you an idea about whether the separation is adequate or not. Of course, when we are going for densitometry, the light based instruments are there. So, source of light also matters depending upon the working principle that we choose, the instrument that we choose, the light source also will vary. It can be as simple as white tungsten light. It can be, if we are going with UV scanner, then it will be mercury or deuterium light, so on and so forth. Now, next comes is uh, the example wherein we are talking about separation of lipids from some biological systems. Maybe say an example wherein we take the example of say E. coli cells. So to talk about the E. coli lipids separation using TLC, here you are seeing the system wherein development system is chloroform, ethanol, and triethylamine with water in the ratio of 3 is to 3.4 is to 3 is to zero point 0.8, right? And the polar, non-polar lipids, etc., are separated on this TLC plate, and the plate is developed using CuSO4, so that you get the chromatogram, and different lipids are going to give you the colored spots like this. Now, as you can expect, as it is the lipid contained is extracted from E. coli, what are going to be the different types of lipids? Yes, mostly it is oleic acid, the analyte A, right? Then it is phosphatidyl glycerol, that is B, is found in lane 5, and phosphatidyl ethanolamine, that is the C, which is seen in the lane 4. All right. So we are going to have these components in E. coli lipids. But when you want to extract these lipids, of course, you should have E. coli cell suspension, which you are going to centrifuge at 800 gram, 800 G for 8 minutes, and then that pellet is resuspended into deionized water or saline for washing. Then the pellet is put into, or buffer also you can be used, and then it can be put into chloroform ethanol water system, so the lipids are extracted. So maximum extraction is done, and that solution of lipids is then vacuum dried, so that, or air dried, so that all the solvent is removed and the dry powder is there, which is then again resuspended into a solvent system, and you are going to go for TLC separation. Right now, when materials like lipid you want to study, of course, other components which are present in the E. coli cell, which are hydrophobic in nature, may also get solubilized into it. Or you have, say, examples like urine sample which you want to fraction it by TLC, or any biological samples wherein there are multiple materials which may interfere with the efficacy of the system. In such cases, what to be done? In such cases, it is required to go for pre-adsorption. So the TLC plates have the area that is about 1 centimeter and 10 by 10 layer, wherein non-adsorbing materials like kieselguhr or paper are there, onto which you are going to directly apply the sample. And these layers are the pre-adsorption layers, which are going to work like the filters to remove maximum unwanted materials, and only the analyte passes onto the TLC plate. So in place of this pre-adsorption methods, you can have the layers which are in continuation with your TLC plate, or you can have a SPE column, right? That is solid phase extraction columns, which are used for preparation of samples. So they will retain most of the unwanted materials, or in simple words, it can clean the extract, and the desired sample can be further taken for TLC. So these SPE columns, they look like this. You have selective columns. You can work uh, for selective columns, in the sense they are derivatized and can be applied for the selective purposes, and then can be used for concentrating the desired extract, and then you are going to take it ahead. Now, coming back to the example of these lipids, when I am going to go for lipid extracts into acidic, non-acidic species, they can be done by Cephadics columns initially, right? It is an example of pre-treatment SPE columns. So, different solvent systems you need to choose one after the other, it will follow, so that you get the desired extract, and then it can be further analyzed by TLC. So it is very essential when we talk about separation of biological entities, that is cleaning out of the sample and then applying it to TLC, so that it will not interfere with the visualization and detection of the analytes. Now here is another example wherein we talk about vitamin E and its derivative forms, oxidized forms. So, why to take this example? Because vitamin E is a known antioxidant and anti-inflammatory molecule, which is going to be having role in maintaining cardiovascular health. So, and the important forms which are found are tocopherol and tocotrienol homologs you will get. So we can check the presence of these oxidized forms as against that of the normal tocopherol, alpha tocopherol. So for this purpose, we can go in for TLC. These are the structures of tocophenol and enols, which differ with reference to the side chain. One is vityl side chain, in another case it is farnesyl side chain, and of course the R groups do they vary. But for separation of the molecules which are so closely related can be achieved by a TLC. So this is an example. The plates are showing this alpha, beta, gamma, delta T and alpha T1. So you can compare the different samples, can analyze and check the presence of relative quantities of the oxidized form and the native form of vitamin E in a given sample. That can also be done by TLC. So it is a kind of a overview we have tried to have about the visualization of TLC plates once they are developed. So thank you.